Webinar will extend until about 7:10 p.m. with any wrap-up questions afterwards. So now I'd like to introduce our presenters. Patrick Edwards is a senior instructor in the Environmental Science and Management Department at Portland State University and director of the Environmental Professional Program. Patrick received his BS in Environmental Analysis and Planning from Frostburg State University and his MS and PhD from Portland State. Gail Shalom is a natural resources specialist and landscape architect and has worked at Clackamas Water Environment Services since 2011. She has over 30 years of experience working in stormwater management, stream restoration, wetland mitigation, NEPA compliance, and low impact development practices. Gail is a graduate of Rutgers University and Oregon Graduate Institute. Glenn Ahrens is an extension forester with Oregon State University Extension Service for Clackamas, Marion, and Hood River counties. And he's the director of the OSU Hardwood Silviculture Cooperative. Glenn has a BS in forestry from Humboldt State University and an MS in forest science from Oregon State University. And last but not least, Dave Bugney is a retired civil structural engineer and incoming board chair of the Clackamas River Basin Council. He holds a BS degree in civil engineering from OSU and an MS degree in structural engineering and structural mechanics from the University of California, Berkeley. Now let's welcome Patrick, Gail, Glenn, and Dave as they present about community-based science in the Clackamas Basin, the contributions of amateur scientists to managing streams in the watershed.
Thanks, Renee. Appreciate you having me on and excited to give my second talk for this series. I'm going to go ahead and share my screen. And are we looking at the presenter view or do I need to switch it?
Need to switch.
Switch. Okay. That should be good, right?
Perfect.
That's it.
Thanks. As Renee mentioned, we're going to be talking about some of the community-based science that's going on in the Clackamas Basin. I've got some interesting results to show you. We're going to hear from Gail about why the county engages in community science, and then we're going to hear about some projects from Dave and Glenn. To get started, I just want to define community science for the group. And community science is a term that we use now instead of citizen science. So, community science is a bit more inclusive. So you will often hear citizen science, and it's still in the literature as well. And we're shifting away from that term and starting to use community science. And community science programs are partnerships between public volunteers and environmental scientists or environmental managers with the general aim of generating data for scientific investigations while also engaging the public in the scientific process. But there's a lot more benefits to community science than just generating data sets. And I like to think about environmental community science as the overlap between the need for data, the societal implications of environmental literacy and environmental health, and how environmental managers maintain or improve environmental condition in their region. There are other major benefits aside from just generating data. And for example, environmental literacy is an important one. A STEM education is a very important outcome of community science. We know that when students are engaged in authentic scientific experiences, they're more likely to enroll in STEM programs and stay in them. And this is particularly true for underrepresented communities in science. We're always looking for civic engagement and for folks to be aware of the projects that are going on in their region and supporting those efforts to improve the environment. With that, I'm going to let Gail talk briefly about why the county engages in community science, and then I'm going to tell you show you some data and tell you a little bit of background about the community science work I've been doing in Clackamas County. So Gail, go ahead.
All right, thanks, Patrick. Well, first of all, Water Environment Services is a wastewater, stormwater, and watershed protection agency. So we have a few different reasons that we like to participate in community science. And some of the projects that Dr. Edwards is going to talk about are things that we've partnered on together or they've taken place on our properties. So the first reason that we are interested in community science is to engage the public and, as Patrick already said, increase environmental literacy. We have a new communications roadmap that aims to better engage and connect us to our customers, and community science can be one way to accomplish this. We hope that community science projects will help the public understand our work and hopefully increase interest in our work, and also encourage the next generation to build on our work and continue our work and build the skills that we'll need for a future workforce. And next slide. Another reason that we participate in these projects is because we have a municipal stormwater permit called an MS4 permit, or municipal separate storm sewer permit. And this permit has a lot of different requirements, but one of those many requirements is to do some public education and outreach related to stormwater. And the purpose is to inform the public about the impacts of stormwater discharges on receiving waters, and also inform them about actions they can take to reduce their impacts on runoff or on stormwater. So again, community science projects could be one way to help us meet that requirement and get the public to understand, by doing hands-on projects, the link between their actions and water quality. And then the third reason that I wanted to point out is that in an effort to improve watershed health, WES does construct stream restoration projects. And we're always learning from every project and try to apply what we learn on one project to the next one. And sometimes even data collected by high school students, as you see in this photo, and that Patrick is going to tell you about, can help us. These were some students doing some measurements in Rock Creek, where we did a restoration project. So we've teamed up with Dr. Edwards and Portland State University for most of the community science that we've done so far. And I will pass it on to Dr. Edwards to tell you about those efforts.
Thanks, Gail. A perfect segue. Before I get into the data, I just want to provide a little bit of context and background information about the work that we're doing, because I think it's important to see how these projects develop. And we did not intentionally set out originally to monitor restoration or to generate these data sets for management. It all started actually as part of my efforts to develop good curriculum. So in the late 90s, when I started teaching at Portland State University, I really wanted to engage my students in field trips where we would go and collect data on streams, and then have a data set for the students to work with for their final projects in the class. And I was looking for just anything we could do that, one, would fill the time during a field trip nicely, and then would generate these good data sets. And so I became aware of something called bioassessment. And when I started teaching, I never had actually even heard of that term before. And bioassessment is using the organisms to tell you something about the quality of the environment. And about 5 or so years after I started working on this, developing this curriculum, I had a student named Daniel Bedell who got really interested in bioassessment. He became my graduate student and now my colleague, and we work together. He's faculty at in the environmental science department. And I just want to mention that Daniel has probably collected about half of the data I'm going to show you. So I think he's on the meeting today. So that's a young Daniel Bedell. So what is this bioassessment? Well, you can use all kinds of critters to tell you about the quality of the environment. What we're focused on is using the macroinvertebrates, or the insects or non-insects that live in streams, to tell us something about the condition of the stream. And there's lots of advantages to using macroinvertebrate bioassessment. Probably the most important advantage is that they live in the stream and they respond to changes in the stream structure and stream health that we may not be able to detect with traditional chemical measurements. So the major taxa we're going to see are from the orders mayfly, stonefly, and caddisfly, and we call those EPT, and those are the short for the scientific names for those organisms. And in a healthy stream, you'll see 70, 80, 90% of the community of organisms come from these three groups. As the stream experiences pollution or disturbance or other factors that degrade the health of the system, the community begins to shift over to more pollution tolerant organisms like snails, isopods is in the top of the picture there on the right, and dipterans, which you may be familiar with. That's a black fly larva. So what we can do is we can go out into the streams, we can measure we can collect the insects, we can measure them, count them, and then infer the health of the stream using these insects. So when I started trying to develop this curriculum, I wanted to make sure that I developed a methodology that did not kill insects. And the reason for that was really just because it was just kind of awkward to go out with students and talk about the integrity of streams and the health of streams and how cool these insects were, and then kill literally thousands of them as we preserve them and then would count and identify. So the first challenge I ran up against when developing this curriculum was the fact that none of the field guides had pictures or descriptions of live insects. So the first thing we did was develop a field guide that was specifically focused on identifying insects with the naked eye in the field without magnification, and based on the characteristics of the live insects. Then I needed to develop a sampling protocol that did not include preserving insects, so we could actually count them and return them to the stream. So I developed this non-lethal method, and what we do is we go out in the stream, we use a D-net, and we collect 3 samples from the stream. We composite those samples into a tub, and then we pour those tubs into this plastic beading tray and randomly select insects out of that beading tray to count and identify. And this is an important step. This random selection helps ensure the quality of the data and the reliability of our findings. So then the students would identify and count the insects. Their counts and identifications are verified by an expert, and then we return them to the stream unharmed. And so does not really harm them at all. So I had early in my career, a pretty remarkable run of consistent teaching. And I went on the same field trip pretty much every fall and spring for over 15 years. And we were going to Balch Creek when we first started. So about in 2005 is when I kind of got the methodology all dialed in, and we started collecting data that would generate reasonable results. And it occurred to me after about 10 years or so that I was actually generating data sets that could be very important to help scientists understand the health of streams and help managers understand the condition of their streams and perhaps even evaluate changes in management practices. So I'm going to show you a couple of graphs like this. I just want to go over a couple of key points of these graphs so you can understand them as I show them to you. So on the y-axis is the percent of the community of organisms that are from the mayfly, stonefly, or caddisfly groups. And that's just one way to measure the community of organisms. There's a whole bunch of different ways, and this is the simplest one and one that's pretty reliable. The other thing I'd like to point out is that each one of these points on the graph represents 1 to 2 field trips, at least at Balch Creek, and about 30 to 50 students. So you can see over the years, I think when I did the math on this, it's about 1,000 students that sampled insects at Balch Creek. In about 2015, I believe, or maybe a little bit earlier, Dan Bedell began taking over these field trips, and he worked with students after that. So when I started looking at this data, I realized that the data is actually telling an interesting story. So if you notice there, I highlighted these 4 data points on the right-hand side of the graph. I want you to notice how those line up with each other. So the variability in the data set is certainly there, but you can see that the students are tracking some change in the EPT community over time. And this is astonishing to me when you think about the fact that each one of these dots represents a different group of students who, for the most part, have never collected aquatic insects before. So when I really started thinking about this data we were generating, I realized that we had some very good data for environmental scientists and managers to use. So we published an article back in 2016 in PLOS One about the value of long-term stream invertebrate data collected by community scientists. And at the end of my talk, I'll upload all these papers for you if you want to take a look at them. So I'm going to talk about a couple of different sites, and I just want to give you a sense of place here. So Balch Creek is up in Forest Park in Northwest Portland. We also sampled in a couple of other creeks, 3 creeks and Clear Creek and Mount Scott Creek. And I'm going to show you data today from Rock Creek and Carly Creek. So in 2014, Gail and the county planned this restoration of Rock Creek. And it occurred to me that this presented a really good opportunity for us to study the impact of the restoration. So I have been collecting data in Rock Creek since about 2010, I believe, and the restoration was planned. And so I recognize that this would be a unique opportunity for citizen science or community scientists to contribute to the management of Rock Creek and to document the restoration of this site. So the restoration proceeded. This is on the right here, you can see they put in a lot of woody debris. They've slowed down the stream. They've reconnected it with the floodplain. And we would expect the insects to indicate something about that change to that system. So we began to look at this data, and I realized at this time that we had this really unique opportunity to look at these data sets from what's called a BACI perspective. BACI just stands for before, after, control, and impact. And this is the gold standard for kind of investigating changes to ecosystems. And the reason why we need this BACI design is because not only do we need the baseline data before the restoration is done, we need the data for after the restoration is done, but we also need a control to make sure that the patterns we're seeing in our impacted stream, in this case, the restored stream, are not due to changes in climate, because the invertebrate, as all biologic populations are, they respond to kind of broad scale climate conditions. So when we began looking at this data, I was just astonished at what the students could observe in the data they collected for Rock Creek. So you can see here, we've got the before construction, the red square is the construction. We've got the after construction. And then the black line is Rock Creek, that's our impacted stream. And then we've got Balch Creek, which is our control stream. So you can see here, after the Rock Creek, after the restoration construction, the percent EPT for Rock Creek really went down. And then notice it's recovering back over 2 or 3 years. And again, notice the pattern and the relationships between the dots. To me, this says the students are actually measuring a real change in the community, and that the data are reliable enough and accurate enough to show that response. We also looked at a whole bunch of different other kinds of organisms. I'll just show you one chart here. This is the insects that are tolerant to the kind of disturbance that took place during the restoration. And notice again, they increased significantly after the restoration. And then you can see again, those populations going back down and coming back into equilibrium with the before conditions. So when I looked at this data, I realized that community scientists have a important role to play in designing these and contributing to these BACI designs, which are so critical to understanding the impacts of management activities, but rarely implemented in the world of environmental management, and actually in science as well, because they're just so difficult to generate these data sets and expensive. So we published this paper in Restoration Ecology, and I just want to point out that my co-author was Gail in this paper, one of our community partner. This was in 2018, and again, as I said, I'll provide these papers for you. So let's look at where we are on Rock Creek. So this is from 2010 to 2021, and to me, and I think for anyone who's in the business of collecting ecological data, this is a pretty remarkable data set. You can see that Balch Creek, our reference site, has stayed pretty much the same for about half the time we've been sampling. And in the last couple of years, it looks like the percent EPT is starting to decline a little bit. And take a look at Rock Creek. So Rock Creek, we saw that collapse in the percent EPT after the restoration construction. And then you can see that the community is beginning to come back and is generally increasing over time. And now is at this, for some of the samples, is at the same level of Balch Creek. And you can see what I think is very interesting here is this kind of variability in the data is showing that the system is still in a little bit of disequilibrium from the restoration construction. But in the last couple of years, it looks like the stream is beginning to stabilize, and we're seeing a general increase in the EPT in Rock Creek. So I was working with teachers at Clackamas High School, and we really wanted to develop some follow-up experiments to these bioassessment studies. So we worked for a couple of years and developed, I think 8 or 9 in-depth experiments that students could do to manipulate conditions in the stream that would actually illustrate the mechanistic processes related to how insects respond to restoration. And here I just want to point out my co-authors were Rod Schroff and Angie Schroff, were teachers at Clackamas High School, and the student Megan Colley. So I want to go back to what Gail said at the beginning of our talk about why students, why the county involves itself in community science. So let's look at some of the numbers. And I really like looking at these kind of summary statistics. So for about 10 or 11 years, we've been working out in the county. It's somewhere between 4,000 and 6,000 students, which is amazing. And I'm sure that's an underestimate, because that's the 4,000 to 6,000 students I could find records of. And I know there's some students out there, some classes out there that I missed when I was looking through my records. We went on more than 80 field trips around the Clackamas County. And this also includes Balch Creek, by the way, to collect bioassessment data on those streams I showed you on the map. And then we had about 30 field trips where students conducted those in-depth experiments. 2 middle schools, 5 high schools, I think about 11 teachers. And my favorite stat to show is it was a cost about 20 or 30 bucks a student to have these really high quality experiences. We also assessed student learning, and this is some kind of typical results from our high school assessment. So now we're getting at, are we increasing awareness in environmental literacy? So these are percent responses, correct responses, or awareness from an assessment we did. I think this is probably around 150 students. So before participating in this program, almost no students were aware of a restoration site, and then 80% or more could name a restoration site in the county. One of my favorite outcomes is we asked students, have they ever discussed restoration or water pollution with someone else? And only a few students had actually ever discussed restoration with a friend or family member. And then after the project after they participated in the project, about 40% indicated that they'd had a conversation with a friend or family member about restoration. And this is like a multiplier effect for the county. So for every student that participated, about 40% of them helped their friends and family become more aware. And then the other thing we did was we had students look at a couple of insects and identify them. In the pre-condition, about 20% of students could correctly identify the insects, which surprised me because we thought that most students would not be able to. But then we realized that a lot of these students had gone on these field trips kind of earlier in their career or in middle school, and were still familiar with the insects. And by the end of the program, 80 or 90% or so could identify 3 insects. So I feel like we really increased the awareness and literacy of students in the Clackamas Basin. One of my favorite examples of a public engagement is when we stormed, so to speak, the Board of Commissioners meeting in 2017. And the students attended a Board of Commissioners meeting and symbolically voted with their scientific posters to continue funding for this program and for watershed health in the county. And my favorite comment by the commissioner, she said that they hadn't seen this many people at a Board of Commissioners meeting since they wanted to expand TriMet out into Clackamas County. So with this public engagement piece, Gail and I published a paper in the Society for Ecological Restoration in 2021, talking about how we can engage students in restoration and the kind of the community science aspects of our program. So what are we working on now? So Daniel Bedell and I and a bunch of other people have been working with agencies in the Puget Sound and the Willamette Valley to develop a metric that is specifically for community science generated data that can help us understand the health of streams. And we're calling this the Community Science Index of Biotic Integrity. The research is finished, the manuscript is finished. We're going to submit it any day now, so hopefully that'll get published. And you can see the metric responds quite well to increasing disturbance in the watershed. And I just want to point out one creek that I think is really interesting. If you'll notice there, that outlier in that chart is Rock Creek. And that outlier tells me that Rock Creek is in better condition than it should be given the amount of disturbance in the watershed. And I think that's evidence that the work that Gail and her colleagues are doing is keeping Rock Creek healthy in the face of actually what is a lot of increasing development in that watershed. The other place we're working is in Carly Creek. And so here we have a data for about 5 years, 6 years. We've got the, in this case, I'm showing the Community Science Index of Biotic Integrity score on the y-axis. And you can see that Carly Creek, after the restoration is the black line. The community score went down, which we would expect in response to restoration, but that community is slowly improving. And so we're need to collect some more data here for another couple more years, but we plan to continue working in Carly Creek to monitor that response to that restoration there. So I just want to take a quick moment to acknowledge a lot of people who worked on this project and helped us generate data with students at many high schools in Portland State University. Got a lot of teachers there, including, I just want to mention Angie and Rod Schroff, who have really helped us develop this curriculum and have been participating in this program for a long time. And then a bunch of volunteers. These are mostly a bunch of students of mine who have come on the field trips and help students collect insects and identify, count and identify the samples. And with that, I think we're going to take questions now, or how would we like to do this?
I think you could take some questions now, Patrick, since my talk will be a little different.
Okay. Let's see. So an IBI versus how do you measure disturbance? That is the amount of watershed that is urbanized or agriculture. Good question.
Do we have any more questions in the chat? If not, I have one.
I just see the one from Ian.
So I'm curious, Patrick, or anybody else who's on the meeting who's worked with these students. I know what high school students are like, and sometimes, you know, they can be really engaged, and sometimes it's a little hard to get their attention focused. So I'm curious, how did you manage the quality control aspect with these groups?
Well, we do some training before they go out. We do some training while we're there, and we just really keep a sharp eye to make sure they're collecting the data, the insects in the right way, that they've done the subsampling procedure correctly, and then I myself or the volunteers verify their counts and identification. So the only place where things could potentially go wrong is in the collecting part, but the randomization, the method itself deals with that a little bit because we randomize which insects we select.
Thank you. It's really an awesome effort, awesome data.
Thanks. Thank you. Glenn is asking, can you explain the IBI a bit more? I noticed it dropped for Balch Creek in 2019, similar to Carly Creek. Yeah. So the index of biotic integrity is comprised of 10 different individual metrics. And I can send you that stuff, Glenn, if you'd like. One of the things about IBIs is that they are affected by climate. So you're going to have variability in the IBIs simply from climate. You also have variability from associated with the collecting method itself. And that's with any metric that you have. For now, as far as we can tell, the index of biotic integrity that we've developed for community science is better than any other metric that exists out there. And that's only because all of those other metrics were developed using preserved specimens, where the people who collect the samples preserve the insects in the field, take them into the lab where they do the randomization and the identification. So that generates a whole different data set than you would see from the citizen science, the community science groups. So we are designing an IBI that is specifically to be used with the kind of data that we're generating in these projects. We have done programs in elementary schools. It's a little bit more tricky to get out into the field with elementary schools, but I mean, I can probably think of 20 or 30 times where I've brought insects into elementary school classrooms and had them learn how to identify mayfly, stoneflies, and caddisfly, which is really fun, actually. Okay, shall we continue on with the next talk with Dave?
Sure. Go ahead. If you
Okay.
wouldn't mind bringing up that screen, and if you wouldn't mind running the slides for me, that'd be great.
Yeah. Are we good with the slides now? Can everyone see?
Yep, I see your last one there. There we go. So thanks, Patrick, and I'm Dave Bugney, and I'm going to present briefly about two community-based science projects I'm involved with here in the Clackamas and why they're important. Our family lives out in the Tule, so I'm going to turn my camera off because I think it'll help with our bandwidth. So just bear with me during my presentation here. So what I'll be talking about tonight is the Clackamas River Basin Water Temperature Modeling and Monitoring Project, which you see a depiction of there on the left, and then the Eagle Creek Watershed Fish Habitat Barriers to Fish Passage Update, which you see on the right. And the photo on the map on the left, I'm working with the Clackamas River Basin Council on that project, and then the other project, the Eagle Creek Watershed, with the Friends of Eagle Creek Watershed. So next slide, please. So if we talk a little bit about the Clackamas River Basin Water Temperature Modeling and Monitoring Project, water temperatures, as many know, in riverine systems play a critical role in the viability of aquatic life, particularly cold water salmonids. And it's often said that other than the presence of the water itself, the water temperature is probably the next most important factor. And so it's good to get a handle on that. And with climate change, it's even more important. So that's why we decided to undertake this. So this information then can be used to identify actions to protect and restore, where possible, thermal habitat and cold water refuges. So to give you an idea, if you look at this Google Earth photo in the lower left, it's hard to see, but you can kind of see kind of a sinuous band there going through the middle of the screen. That's Clear Creek near Viola. And you can see there's not much of a riparian buffer on either side. In some spots, hardly anything. Now there's maybe 50 to 100 feet wide. And that's important because if you look at the figure on the right, the stream reach characteristics, the amount of heat that is impinged upon the water, whatever it is going to be buffered in some way by the riparian buffer width that you see there in that figure. So if you imagine, if you don't have any buffer there, there's really no way to shade the stream in essence. And so obviously as the stream gets larger, the buffer effect diminishes, but nonetheless, buffers are very important. Next slide, please. So for our water temperature project, the goal is to create a basin-wide geospatial stream network analysis of water temperatures. So the data that myself, as well as other volunteers, are gathering, which I'll get into in a little bit, will be analyzed in a spatial stream network statistical analysis depicting present day and future conditions, utilizing 3 climate change scenarios that are found in the IPCC report. And that'll be integrated then, that PSU study, into a larger ongoing volunteer-driven water temperature project, which we're leading, which is supported solely with in-kind donations of labor and materials from many sources. So if you look at this little concept here, you can see this is just a thermal gradient map showing you basically where waters may be warmer and where they may be cooler in the Portland area. Next slide, please. So for this project in particular, if you look at the map on the right, that's the Clackamas River watershed. And the red triangles are water temperature or data loggers, sensors that are deployed by the agencies, for example, the Department of Fish and Wildlife, U.S. Forest Service, USGS, et cetera. And then the yellow stars are the sensors that our group of volunteers put in. And you can see the ones we were responsible for were mainly in the lower Clackamas. So this project is really twofold. One is for us, was actually to deploy these sensors and then analyze that data. But then secondly, it was that the temperatures that were recorded by the agencies typically are just used by the agencies themselves. There's no central clearinghouse to share that data. So we are acting as that at this point to kind of amalgamate all this data together. So there's about 80 sensors basin-wide last year that we had put in. And as you see the photo on the left there, there's about 80, or not in that photo, that particular cooler there, we have about 35, 34 or so that are in that cooler. I'm checking how accurate those sensors are. But when they're placed in the water, they're taking temperatures every 30 minutes. And they did that from about June 19th through September 30th. And the other thing that's really cool about a project like this is Patrick was alluding to how to get other folks and just your volunteers involved, is that most of the locations where the yellow stars are were on private property. So I had to get permission from all these landowners to come onto their place, put in these sensors in the water that flowed through their place. And so that, of course, they wanted to know what we were doing and all that. But once we explained it to them, they were basically saying, well, how can we help? Because once they understood what we were doing, they appreciated the value of the work that we were doing. So in essence, like Patrick's saying, you're helping to spread the word through the projects like this of how important the data that you're collecting and then the future results from the data analysis that's going to be occurring really is. Next slide, please. To give you an idea what we did in particular, if you look at the photo in the upper left, that's me in our garage. So just, nothing fancy here. And the data loggers or the sensors are in the left or the right side of the table, of that fold out table I got. And then they're also in front of the laptop. But I just took some scrap PVC pipe and bought some fittings for them. And then the sensors get installed in those housings, those protective housings. And then the sandbags on the left side of the photo are used to, as you see there in the photo on the right, I put the sensor inside its protective housing in the sandbag. Then we also put some boulders, or not boulders, but cobbles and gravels in that sandbag to weigh it down so it doesn't flow away when there might be some high water. So if you look at the photo in the lower left, you can see what the condition looks like when it's in service. So you can see there's the green sandbag. You kind of see the outline of the protrusion of the housing with the sensor inside of it. And then the further weighed down with more rocks just to keep the bag in place. Next slide. So the raw data that you get out of this, to look at this graph and explain it a little bit, on the abscissa or the x-axis there, that's the last summer. So you can see where the data started to be taken around third week in June and extended out just beyond the first week in October of last year. And then on the ordinate or the y-axis there, you can see the temperatures as recorded in degrees Celsius. And what's interesting when you look at this is we captured just by serendipity, we managed to get the sensors in before we had that very anomalous heat dome last year. So you can see how quickly in this case, this is Clear Creek upstream of the Carver boat ramp, how quickly it responded to the temperature increase due to that heat dome. And the temperature spiked at a little over 30 degrees Celsius, which is about 86 degrees Fahrenheit. So if the fish in the salmonids, the fingerlings or the fry, were not able to find cold water someplace else, they most likely would have died or become very prone to bacterial diseases and that sort of thing. But the other thing that's interesting when looking at this graph that you can see is how much the water temperatures in the water fluctuate on a daily basis. So each of those jagged peaks and valleys represents one day. So you can imagine as water cools off at night, when the sun goes down, the temperature drops. And then as the water temperature increases during the day, as the sun comes out, the water increases. But you can see how it can vary by maybe 6 or so, 7 degrees Celsius just during the course of a typical day. So it's pretty impressive. The next slide, please. And that's essentially where we're at with the water temperature project. The PSU now has all our data. They're starting to create their model, and we should start seeing some results probably next year. But we're already getting ready to deploy the sensors for this upcoming summer, which will be happening in the next couple of weeks. So we're excited to keep the project going. Now, the next project I just want to talk about is the Eagle Creek Watershed Fish Habitat Barriers to Fish Passage Update, which basically means that the Oregon Department of Fish and Wildlife has an inventory of barriers to fish passage. And those of us who are into looking at studying fish know that particular salmonids like coho, salmon, chinook, steelhead, all that, they migrate upstream, to spawn. And then when they get to a certain age, when they're ready to smolt, they need to migrate downstream and go out in the ocean. Well, if there's barriers inhibiting them to do it, it reduces their available habitat, right? So what we did here was a group of us from our community went out and we walked all the creeks in the Eagle Creek watershed, and it's about 60,000 acres. So it's a pretty large watershed. It took us a little while to do, but what we did was the barriers that I've outlined or identified with particular nomenclature, like EGB06, for example. These are all barriers that the Department of Fish and Wildlife had either noted that either have changed for some reason. Either the barrier is no longer there, it's still there, but it's worse off than what they thought, or there are some new barriers that they didn't catch that we found, and we've annotated or identified them accordingly. So what I then did was, we created a table with accompanying photographs identifying each of these barriers that needed to be reflected in an upcoming database update. And these are very important because these barriers drive, at least they should if you have used wisely, where one would do fish habitat restoration work and this sort of thing. So next slide, please. So this is the last slide, and I'll just spend a couple minutes on this one because it's kind of interesting. So the types of barriers, you might wonder what we're talking about, and generally they fall into several categories. If we start on the upper right and go clockwise, you'll see culverts. So this is an example of a culvert on a logging road up near where we are. It's a 4-foot diameter culvert, and you can see how there's no way fish can get up into this culvert. And Department of Fish and Wildlife has this correctly identified as a complete fish barrier. There's an insufficient pool for them to even try to jump up into the culvert. That's about an 18-inch jump, which a salmon, it usually has no problem making an 18-inch jump, provided the pool at the toe of this culvert is deep enough for them to get enough, hydraulic force to get up there. But then the other problem you can see, and this is you can see from the moss growing on this wood, it's been there for a while. There's this additional problem that the fish have to surmount. So this is a barrier to an anadromous fish stream, which is unfortunate. Now, if we go to the photo in the lower left, that's a falls in a creek in our watershed, and it's one of the more amazing falls in the Eagle Creek watershed. This is on a tributary, but you can see that it's a step falls. Its overall height's about 13 feet. Department of Fish and Wildlife has it indicated as a complete fish barrier, which I would probably agree with, but there may be some potential flow at some point where a fish may get up because of its step nature. But the problem is, again, it's got an insufficient pool at the toe of the falls in order to make this jump. So most likely, probably 90% of the time, it's a barrier to fish. But then coincidentally, just about maybe a quarter or half a mile upstream of this falls is this, in the upper left photo, is this infrastructure, this concrete barrier, complete barrier to fish. This was developed decades ago. There was a permit for it and everything, but back at a time when people really weren't as concerned as they are today about these things. But there's no way fish can get up beyond this, and the owner that built this even admitted they've never seen any fish beyond this. And just to kind of close out the story is that a number of years back, I don't know, maybe 10 years or so, the county decided to replace some culverts farther upstream of this concrete infrastructure. So they put in a bridge, which is, okay, great. But they put up a sign saying that this bridge was now they removed the barrier to fish passage for this creek. Well, had they bothered to study the data, they would have understood that there was no way the fish could get up there anyway. So in a nutshell, this indicates why studies like this are important. You don't need to be a PhD in any particular scientific field, as Patrick's mentioned, to undertake these things. Projects like this are very important because our state government and even local governments don't have the resources to do all the work that needs to be done, and you can do something really cool for your community that has a definite impact. So with that, I will end my talk. I don't know if there's any questions, Renee or Patrick.
Thank you, Dave. So I do have a question. I remember that you were reaching out to students at the college and people elsewhere to involve them in this effort. And I wondered if you have plans to continue doing that, to try to involve more people in the actual placement of the sensors or collection of the data.
Yeah, I think that would be a good thing, especially in the lower parts of the Clackamas River watershed, closer to Oregon City, for example. I would love it. I think if they want to just forward me their email address or the contact information, and I can talk to them and see if they're up to it. And if so, that would be great.
So if anybody here on the meeting tonight is interested, they can just reach out to you, it sounds like.
Correct. And they can contact the CRBC if they need to, and they can give them my email address. But I think the more citizens that get involved in these kind of projects, the greater they can appreciate them, and thus the greater the chances we can make some profound effects in how the problems that we're seeing with these studies.
Okay, thank you. Do we have any other questions from anybody?
I have a question for David.
Sure.
With the fish passage monitoring, how is that feeding into the plans with the Clackamas River Basin Council looking at fixing that?
Yeah, good question. At this point, nothing formal. We haven't done anything formalized yet. What I figured the steps would be is that I've given the information to ODFW and their GIS group, and they will update the maps accordingly. And then anybody's free to use them for any of these kind of things. But I think it would be helpful for the CRBC or any group that's anticipating doing any fish habitat restoration work to make sure that there aren't any barriers, downstream where you want to do the work, so that you could maybe allocate those very scarce funds elsewhere.
Okay, any other questions for Dave? And if you think of something, we'll have more opportunity for questions at the end of Glenn's presentation.
Thank you.
All right, I think we're ready for you, Glenn.
All right, very good. I will share my screen. I hope that it works. All right, very good. Thank you. All right, well, I'm going to talk about a program that I've been involved with OSU Extension and a lot of partners and community members. We call it the Oregon Season Tracker. And, the overall focus of this is to engage people in climate research. So we're looking at monitoring weather patterns and specifically rainfall, precipitation, and also plant growth, or the stage of plant development, which is sensitive to climate variability and climate change. So that's a program that we started back in 2014, and I'm just going to launch into that. It's a statewide program, but there's certainly good application, and we'd like to see more participation, focus on the Clackamas Basin that might be useful, locally for looking at some of the, the trends and what's happening, finger on the pulse of climate in our local area. So it's a joint project. We started with OSU Extension, and our idea was teaming up with the researchers who might actually use the data, because as Pat was explaining, the connection between the community members who are collecting the data, really want it to be a useful source of data for the researchers for looking at some of the issues and problems or phenomenon that we're studying, as well as, of course, the other aspects of engaging people in science and science literacy. We look at, particularly in the case of our study or our project, is the H.J. Andrews experimental forest, which is down in the McKenzie Drainage and Blue River, where there's an awful lot of very focused forest and watershed research looking at climate and plants and seasonal development. And also then the PRISM Climate Group, which is really an important hub for climate science and also weather data, and expanding their ability to look at how it varies across the landscape. So we are looking at engaging volunteers throughout Oregon. And getting a little bit deeper into it, since we started it, we've engaged about 450 volunteers in 22 counties. And we're using some techniques or some programs that are accessible outside of our Oregon Season Tracker, but with Oregon Season Tracker, we really kind of organize and coordinate this connection with these specific research projects and teams at the H.J. Andrews and also at the PRISM Climate Group at OSU. And of course, everybody could participate. So this is a fairly, easy, especially particularly with the weather monitoring, the precipitation monitoring, using a nationwide protocol, and it's fairly non-technical, and almost anybody, could do it and hopefully would enjoy doing it. And then on the aspects of the plants and the seasonal changes in plants, like when they burst bud and when they develop full leaves and fruits and flowers and fall changes, that gets a little bit more technical, but really very useful for understanding the seasonal impacts, looking at the same kinds of plants around the state and actually around the country. So any of you are welcome to join up. I hope some of you will, especially around the Clackamas Basin. And so looking at what our Oregon Season Trackers do, again, we're focused first on precipitation, and I'll explain a bit about the particular rain gauges and protocols that we use, and then also the plant phenology data. And we would then establish, if you were cooperating in this, as some of these 450 volunteers are across the state, it becomes a documented Oregon Season Tracker site, and in your yard or the woodland, or particularly like a community park or farm or ranch or demonstration area. And schools also are really good potential participants. And then as far as more of the the basics of it, again, a fairly non-technical procedure with a really well-established nationwide consistent protocol in the case of both the rainfall and the plant phenology. And then we're focused on, in our case with Season Tracker, we're really connecting at some fairly specific scientific questions of interest. So I mentioned the H.J. Andrews experimental forest. For me, that place holds some special importance. It was my first job as a forester and forest science assistants in Oregon in 1983. I started working on the old growth wildlife habitat project with Tom Spies and Jerry Franklin, and spent a couple of good years at the Andrews studying old growth forests, and then going to Oregon State after that for a master's. So I really love the Andrews, and here's a great example of one of the good long-term research projects related to just monitoring climate. As you might expect, this research forest, this site has a lot of meteorological stations and an awful lot of intensive data collection, looking at all aspects of climate, temperature and rainfall and precipitation, as well as, humidity and other variables. And so looking at what we're doing with the, the statewide Season Tracker project, some of the relationships that they can look at the fine scale on the Andrews, we'd like to look at across the landscape and maybe validate some of the findings and the modeling that they're doing at this fairly small scale of the Andrews, which is about 10,000 acres, and testing that out across the broader ecosystems across the state. I should note that the Andrews is known as a long-term ecological research site. If you've ever heard the acronym LTER, those are distributed across the country and the world. But of course, when you think about Oregon and even the larger Pacific Northwest landscape, we have a wide variety of climates and ecosystems. And so outside of the more moist forested sites here in Clackamas and at the H.J. Andrews, we're looking at the broader landscapes across Oregon and the dramatic, variation in climate. But particularly then some of this data helps us understand not only how different things might be, in different climate zones, like, the dry juniper forest of the high desert, but also just how it changes on one side of the hill or the other, or up and down, even at the local level with some of the fine scale data we can get. And one of the values of working with OSU Extension is that we're our statewide program with a lot of consistency and presence across the state, and particularly here in Clackamas County, I should note, where we have one of the largest extension programs in the state. And I'm really glad to be able to work here with such good support from Clackamas County and all of you. But across forestry and natural resources and agriculture and our 4-H programs, a lot of different aspects of extension are engaged with the community. And it's the master gardeners, for example, are often really interested in this kind of community science. And also the youth programs and working with schools is really important. And I'll touch on that a little bit more later, but that certainly gives us a good connection. That's why we think the season tracker is kind of a good way to engage the community and spread the word about a community science program around the state. As far as exactly what is done with Season Tracker, we are using the COCO RODS is the short acronym for it, but it's the Community Collaborative Rain, Hail, and Snow Network. It's a very well developed network nationwide, and it provides a very consistent protocol. You need to use their particular rain gauge so that the readings are always consistent, and it's used by many different researchers. Here in Oregon, I mentioned the PRISM Climate Group, the Oregon Climate Change Research Institute, National Weather Service, hydrologists, emergency managers. Some of the aspects of this, would allow the observers to note when there's a really intense weather event going on and it's picked up in real time by some of the emergency managers who pay attention, and COCO-ROS is kind of on their radar for that. So we use that for our precipitation monitoring. And then for the plant phenology, when does the growing season begin, from bud burst to leaf expansion to full leaf to fruiting, flowering, and then to fall and into winter. That is really a finger on the pulse of how the plants are reacting to the climate variability from season to season, as well as change over time. And we use Nature's Notebook, which is again a nationwide program, but then we impose our particular focus, which is we selected a certain list of species of 8 native species in Oregon to really intensively monitor those around the state of Oregon. That also then ties with the research at the H.J. Andrews, and also we hope some other across other ecosystems in Oregon. And some of the key questions that we're looking at, the patterns that they're studying at the Andrews and other intensive research sites, are those consistent across a larger spatial scale? Some of the rainfall patterns and how we kind of understand how it varies with topography, or just how maybe how variable it is in general, but more tied to also the specific mountain topography and changes from hilltops to valleys. And then with the PRISM Climate Group, I want to really credit Chris Daly, who's been the mastermind behind the PRISM Climate Group and a lot of the great tools that they provide for putting together climate data and monitoring not only past data, but then looking at how it might project into the future and getting it again downscaled to the local level. So this is again is where, you know, we look at our community science data as contributing to that. And Chris is really tied into that. And he himself is a Oregon season tracker observer, even in his own backyard. He's responsible, the program is responsible at PRISM. You know, they make some of the, you know, the climate zone maps that gardeners use for looking at, you know, climate zones and tolerance for plants, and they kind of redraw those from time to time. And of course, you see that those are kind of being updated as things are shifting a bit with climate change. But some of those are, you see on the, even on the back of seed packets, looking at what climate zone are you in, when should you plant those seeds, and they're using actually this output from the PRISM group. And also then looking at, you know, how the plants are reacting. Mark Schultz is one of the key collaborators. He's a site manager and scientist at the H.J. Andrews, and really has helped us design this Oregon Season Tracker protocol of how we're using the plant phenology, the Nature's Notebook. And he's got a great network of monitoring of many plants across H. Andrews. And then again, we're just looking at 8 species in Oregon that we're using in Oregon Season Tracker. But he's really been great to look at some of the key questions that we're trying to answer. One thing you might look at if you go online, you know, Google the green wave, and this kind of data collected nationwide, you can just see, you know, the progress of the seasons from year to year. You know, when do the willows leaf out? When do the cottonwoods leaf out? When do the vine maples leaf out in the Northwest? That's partly, you know, what we're focusing on. And with the Oregon Season Tracker, just our participants, we're looking at over 40,000 observations have gone into the Nature's Notebook just over the last few years. And so you could add to those if you'd like to join us. And as far as how it works, you know, you can look at just one plant that you revisit every year to track it, or else, you know, a whole series of plants. We'd like to have, you know, a rain gauge and plant phenology on the same site if possible, but even different parts of one site, like higher elevation, lower elevation, across a larger area, like, you know, the Eagle Creek watershed, for example. And then you can get more advanced. You can look at, you know, making some comparisons. You can look at establishing more of a regional subgroup with a public, you know, kind of a phenology walk with schools or with volunteer programs at demonstration areas, you know, really getting quite a bit of a research site established, not just one, you know, one plant, one rain gauge to put dots on the map, but, you know, more of a community site that you get a lot of people involved in helping you with the data collection. And with working with schools in particular, you know, this is a great opportunity. I was really glad to see your projects, Pat. I reminded when I was working out in Astoria, I was the extension forester there, and I was introduced to the EBT and the value of the macroinvertebrates and how great that was as a, you know, a tool, and also working with the youth on that. And we started always incorporating that macroinvertebrate EBT protocol in our forestry field days for our 6th grade tours, and really enjoyed working with the high schools out there to get mentors from the students there. So I really appreciate that. And I think the season trackers and what we're doing is easily adapted to school programs as well. And the students get pretty fired up about it once they get engaged and realize, you know, what where their data might be used by researchers after they, especially when they meet the actual people that are using it and doing the research and seeing some of the results, you know, on the map. And of course, for our own sake, as a participant, you know, you can get a lot of good reports, in this case from the precipitation monitoring, looking at the rainfall data. I was just looking at one of the Kokora stations in Estacada area, not too far from you, David Bugney, and just reminding myself how dry last April was in 2021. We had a lot of problems with our tree seedlings because right after we planted them, April was just extremely dry with less than an inch of rain, and also that warm east wind that we had. And what a difference it was this year with more like 12 inches of rain in April. So there's some benefit just for tracking your own local weather and maybe looking at how it differs just across our area. And I always check the ones between Estacada and Oregon City and further out in Salem and down by my own little woodlot down by Philomath, and see, you know, what's going on with the Kokora's rain gauges on a daily basis if I want to. If you're interested, then it's pretty easy. You can go to the OST website, which I think Cheryl put in the chat. And nowadays we're doing an online training, and then you sign up to be an observer and get your station logged and start reporting using the COCORA's rain gauge in this case. And you can choose to do just the precipitation monitoring, but if you want to really get into the full deal, you would use the Nature's Notebook and do the plant phenology monitoring. And we're really particularly focused on vine maple. If you don't do anything else, if we all monitor what's happening with vine maple, it really helps put a finger on the pulse of how the seasons are developing and how the plants are reacting across a range of sites in Oregon. And then you would also be getting the newsletter and tuned into some of the results that come from the broader COCORA's research family in terms of the rainfall and precipitation, but also in terms of the phenology from Nature's Notebook. And, we've collected some data. I saw, Pat, you had a great example of the impact that you were having and how students were learning. And we've done some of those assessments with our COCORA's and Nature's Notebook observers with Oregon Season Tracker. So I just had end with a few quotes from some of the participants in Oregon Season Tracker, just using it to look at how much water you're getting, and when if you feel like you need to water more in your garden, being part of the research, of course, and contributing to the science, and then advancing scientific knowledge and citizen engagement. And then, of course, some have a lot more nuance and details, really focusing on what they're learning from observing, the changes in when their plants leaf out and how the seasons progress. So that's my brief story about the Oregon Season Tracker. And if there are any questions, I'm happy to answer those.
It looks like there's enthusiasm for participation, including for students, and Maddie may wish to elaborate on a suggestion she was giving.
Okay.
Well, that just is that I'm a resident of one of those new residences in the Rock Creek watershed, and I'm at the one of the entrances to the Hidden Falls Nature Park. And at the other end of the trail is a brand new high school in Clackamas County, the Adrian C. Nelson High School, which is actually on the edge of the forest that is the riparian area for Rock Creek at Hidden Falls. It seems a wonderful opportunity to connect that high school, as OSU seems to have done with Clackamas High School, but connect this high school with the Rock Creek ecosystem and get their buy-in for something, for a long-term project. They're wonderfully poised to do monitoring.
Do they have a good place to install a rain gauge, or maybe identify some plants for repeated observations for the Nature's Notebook? I
think absolutely, both property that looks like it's owned by the school district, but they're also immediately adjacent to the Hidden Falls Nature Park. Students walk through the nature park down to Rock Creek, down to the Hidden Falls, and then back up to the high school on a daily walk to the school. So it's wonderfully poised for them to feel part of that ecosystem. We just have to get them away from their screens, and back down into the Rock Creek Canyon. I don't think it'll be difficult if we show them, provide them with the opportunity for this program. Season Tracker sounds wonderful for this purpose.
Yeah, definitely connect with us about that. And I happen to have some of the official COCORA's rain gauges in storage at our office, because we used to teach it in person. So we do have rain gauges that maybe we could get it to you faster than the ordering online or whatever. I should note that we're really trying to get more rural participants and get a better spread of observation points out in the farm forest country and away from suburbs and towns. So we're really hoping for that. And but certainly Rock Creek would be a great site as well.
Thank you.
You're welcome. David.
Thanks, Glenn. Just a couple of quick points here. I just wanted to reiterate the importance of the COCORA's. We've been collecting rain data at our place for over 30 years and been with the COCORA's system for about 4 years or so, and it's very easy to do. You can do it on your smartphone every morning. It's just after I eat my cereal, I go outside, take my rain data, enter it in there, boom, I'm done. And then the second thing too is that Glenn was mentioning the H.J. Andrews Experimental Forest, and that's a favorite place of mine as well from back in Oregon State days. And one thing I think people might find of interest is that to understand that place better, but there's a book called Forest Understory, and you can get this through the Clackamas County Library System. This is my own copy, but you can certainly get it there. But this is, I don't know if they still do it, Glenn, maybe you know, but in the past, they used to invite writers out and artists out to kind of give you more of a holistic experience than just a bunch of scientific researchers. And what this is it's a compilation of the best writings at the H.J. Andrews Experimental Forest. And it talks about citizens or community-based science. It talks about just the beauty of the place and helps people connect in a way I think that has been missing. So if people are interested in that sort of thing, this is a good book to check out.
All right, thank you. That nothing better than a testimonial and diving deeper. There's so many great stories and books written about the forestry research and from the H.J. Andrews. It really is a great place, and it's not the only one, of course. So, and let's make the Clackamas Basin our own experimental area for a lot more of this community-based science data. Are there any other questions?
I have a question, Glenn.
Yeah.
And I'm sorry if you've already mentioned this, but I'm curious who has access to the data that's being collected through season trackers. Is that available at all to the public? Like, say, for example, if a school got involved, would they be able to look at some of the data so that they could do some additional work with that?
Yeah, I mean, there's a couple of ways. One, the basic data, either CocoRa's or Nature's Notebook, you just access it through their system. So they have a great, you can when you join especially, but I think even someone who's not a member can log in. I'm not certain about that, but best case is to join. Then you can explore the data in many different ways, and you can even request and correspond with the site managers, the program managers, to do more in-depth kind of investigations. So that would hold for both CocoRa's and Nature's Notebook. And then there's the affiliated research that is done using that data, and you can contact the researchers. For instance, the PRISM Climate Group they have a wonderful set of tools that anyone can use. But if you wanted to inquire or get students more involved in doing a study, I would imagine if you corresponded with some of the folks at PRISM Climate Group, that you could get them engaged and kind of dive into a little bit more about how they're using the season tracker data, as well as the broader data that they have, and how do they look at some of these aspects of climate variability. Some of the work they're doing at the Andrews is just fascinating. Chris Daly is really looking at, what does the change in the macro climate from 100,000 foot level, how does that really translate to your local area, whether you're on a hillside or a mountaintop or a valley, and how the weather patterns in the Northwest come with cyclonic or anticyclonic systems that come in. And, that's what really makes the weather that averages out to be the climate. And they, for instance, at the Andrews predicted that, with a 5-degree climate change on the hilltops, it would be more like 8 degrees, and down in the valleys and the cold air drainages, it'd be more like 2 or 3 degrees, just from some of their modeling. And then, whether it's a day that has an inversion or not makes a huge difference in the actual, what you're going to observe. So there's a lot of neat connections to explore getting into that. But I bet you can engage students just using what's out there on the web with those two programs, or you could contact researchers and go deeper.
Okay, fabulous. Thank you. Does anybody else have any questions for any of our speakers tonight? Now's your opportunity.
I just want to mention I uploaded those papers to the chat if anyone would like to download them and take a look at them.
Thank you,
I've downloaded all of your papers, Pat, and they'll be on our website.
Okay, great. Thank you.
Okay, well, if there aren't any other questions, thank you to our speakers, and we'll wrap this up. And before we say so long, I have a couple of announcements. One is that The date for our last session is yet to be determined due to some scheduling difficulties, but it will most likely be either June 7th or the 14th at 6 p.m. And we'll send out notices to all registrants about that when it is decided. We'll be having a presentation about public policy and how it guides restoration and natural resource use within the basin. And this will be our wrap-up final session, tying everything together with speakers who were and are currently in the trenches developing such policies, which gives us some idea why there's scheduling difficulties. You can use the same Zoom meeting link and are already registered for the event. We'll send a reminder email the day before, though. We will send you a thank you email with these links, as well as the link to the recording from tonight's session, and more information about getting continuing education units from Clackamas Community College if you're interested in that. Special thanks to our speakers, Patrick, Gail, Glenn, and Dave, for tonight's encouraging and educational program. You guys are all doing great, amazing work that I know is making a big difference amongst the population that you're touching. I'm especially thinking of those students who are getting out there in the field and doing some real world science. We also thank our gold sponsors, Oregon Beverage Recycling Cooperative and Clackamas Water Environment Services, and bronze sponsors, the Clackamas River Water Providers, Metro, Port Blakely, and the Geological Society of the Oregon Country, who are helping us keep this conference free for all participants with their support. Every little bit helps, so if you like what you see, please consider a small donation. And thank you for joining us tonight. Please let us know what you thought of tonight's program, and we'll see you next time for our last webinar. Take care, everybody. Bye-bye.
Bye.
Thank you. Great job, Renee, and everybody else.
Thank you.
Thank you all. Great presentations.
Yes.
Good.